Now, I'd like to thank the organizers on the record for inviting me today and speaking at this wonderful meeting. It's really great that Bill was first to speak and introduce the general concept of using the tags for affinity purification and, started the whole discussion about that. Now, I will switch gears a little bit because whereas when you're purifying a protein, you're going for the highest yield of the most pure protein and you don't want any contaminants with it. Our approach is when you're analyzing intracellular signaling complexes are actually exactly opposite. We want everything that cell puts on that protein to extract and analyze in the most intact fashion possible because we're interested in the intracellular partners of our protein of interest. Now, my lab uses Drosophila as a model experimental system because we like the flies because we can use genetics and biochemistry and quickly do biological validation of certain things. And now I'll show you that it's extremely important in these kinds of experiment to follow them up with biological validation. So my main interest is in developmental signaling pathways. We would like to know how cells communicate during development of the embryo. In the process, in the past 20 years or so, people have mapped a relatively small number of signaling pathways that are responsible for building the shape of the embryo, for the developmental processes. Some of them are listed here, and I'm sure most of them are familiar to you. And the prevailing paradigm has been that a certain signal is received outside of the cell, and then the receptor reacts to it, transduces the signal to the intracellular signaling pathway, and then somehow it reaches to the nucleus, and there are target transcription factors that will activate batteries of target genes, and then that summed up results in developmental processes and cell fate choices. Now, these days, I would say that this view of linear signaling that happens from the outside into the cell and then has an outcome is rather simplistic. And we like to look at cellular signaling through the prism of a network of interacting proteins. Because a protein never works in isolation in the cell. And it has all these binding partners. By the way, this is a protein interaction network of C. elegans that Mark Vidal's lab produced some time ago. But we like to think that signals come in from the outside, and then they go through this complicated network of protein interactions. And the final outcome is very much dependent on the kinds and the functions of these protein-protein interactions. And this is exactly what we would like to find out as it applies to developmental signaling.